had a really good morning talking to a few of you so far. Looking forward to the rest. I'm not sure the nature of these talks usually, but I'm really happy for it to be informal. You know, I'm hoping to motivate discussions about how any of these tools might be interesting, useful to things that you might be thinking about. So, you know, I'm good having you interrupt me if you have a question. So yeah, I want to tell you about some of the tools that my group has been developing to organize cellular interactions in potentially complex communities, looking hopefully at both adversarial and synergistic interactions that might occur. A lot of our work has really focused on looking at microbiological systems and trying to understand the transitions that happen into group behavioral phenotypes with small cellular ensemble numbers. So on the left here is, it's actually a three-dimensional box. It's a little hard to tell. We have an inner cavity of Staphylococcus that's surrounded by a shell of Pseudomonas. This is a 10 micron scale bar, so you get a sense. And these sorts of structures have allowed us to start to ask questions about how the behavior of these different populations change in proximity. What you can't tell from an image like this is the nature of the material that we use to segregate cells. I'll get into that in some detail, but basically these are protein hydrogels that we're micro 3D printing. They are able to isolate cells while allowing facile chemical transport through the walls. So we can grow very high densities of cells, for example, because we can get nutrients in, we can get waste products out, and we can look at chemical signaling that happens through the walls. We can do other fun things. So on the right side, let's just kind of a cute demonstration of how we can guide bacterial motion to create currents that can do things like move objects around. And we've used this phenomenon to do things like drive flow through tubes. So, you know, I think probably many, if not most or all of you have done cell culture at some time and are aware of these amazing developments, amazing revolutions that happened in cell culturing. First from Pasteur, who developed liquid cultures, could grow large amounts of bacteria to study. And then Robert Koch, actually Petrie was a scientist in Koch's lab, developed the Petri dish, and this really allowed people to start identifying individual bacteria. You could grow solid colonies and identify the source of different different diseases. As transformational as this technology was, it was 19th century technology. Really, you know, we're living in the 20th century. We understand so much more about how cells interact. All right, so there are many developments that have occurred, and the technology that I'm going to tell you some about has provided new capabilities in some of these areas, along with a range of other micro fabrication techniques. In many instances, if you want to study how small numbers of cells come together and alter their behavior, you'd like to be able to confine cells in volumes that allow them to grow in these small ensemble groups. And so this very schematic picture is showing something kind of similar to what I had on the title slide, where we might have one chamber that's a picoliter in size that contains some number of cells, compartmentalized and surrounded by another group of cells. And they may have very different behaviors. They may have different types of signaling molecules. But one of the key parts of this, as I mentioned, was being able to both confine cells in these very small volumes but also to fabricate the materials in a way that allows the cells to grow, to exchanging waste and nutrients, and also potentially to allow signaling molecules to be able to move through. This slide should have advanced, but if you can strain a little bit, this is a transmission electron micrograph showing one of our materials. It's multiphoton photocross-linked bovine serum albumin. It's a protein that we typically use for a variety of reasons. It is very soluble, so that's one great aspect of it. It's easy to undergo photochemical reactions involving singlet oxygen. And it's really cheap. And here's a 200 nanometer scale bar. And what you can hopefully glean a little bit of here is that it's highly porous. And by altering the fabrication conditions that we use, so laser power, laser scan speed, different parameters involved in the fabrication, we can alter how porous these membranes are. So what this should show you is that we can tune the parameters of our materials to allow some molecules to be able to diffuse through. So signaling molecules, for example, from the circular cells inside the core to get through and interact with the cells on the outside, but to create conditions such that another type of signaling molecule, such as those contained in the pink triangles, not to be able to transmit through. As I showed you on the initial title slide, we have the ability to control motility of cells by creating structures that guide them in certain ways. Again, on microscopic scales, we're able to organize communities of cells within 3 dimensions. What we fabricated in this case are 2 different types of cells. So one is it's a raw 264.7 macrophage cell line cell. And we've been able to combine that mammalian cell inside a chamber that also has a box of high density Staph aureus inside. And we can observe, for instance, the macrophage attacking this little cube of Staph aureus, eating its way into the box, sucking up the Staph aureus, and then blebbing out, dying as a result. It's a very cool movie. I apologize that you don't get to see it, but maybe you trust me. Our technology also allows us, for example, and this is one of the things we were talking about this morning with some of you, but to pattern chemical gradients, to do things like localize cells to different regions. We've worked with neuronal type cells to guide regrowth of peripheral neurons. So this technology generally involves using biotinylated bovine serum albumin and then using an avid and biotin display system. We also, with these protein-based hydrogels, can make materials that have very different stiffness properties, so very different Young's moduli. We can make materials that range from maybe about a kilopascal up to a megapascal. Those of you who are working with mammalian tissue, this sort of runs the gamut from the stiffness of brain up to the stiffness of connective tissue. And that's really nice. It's really valuable if we want to create environments that better mimic the biological home of the cells that you're trying to model in situ. And to give you a sense of where some of our interest has come from in our work with Marvin's lab, the microbiologist, with bacterial cells, if you're not in the field, I think most people think of bacteria as being sort of lone rangers. They act on their own. They're single cells. And in fact, they have very rich social behaviors. They develop phenotypic changes that, for example, allow them to become much more pathogenic, virulent, Many of you may have heard of the term quorum sensing, where different genes are upregulated once the cellular densities become high enough. So the cells secrete signals and tell other bacteria that they're there. This phenomenon obviously is not unique to bacteria. So this is a Wisconsin football team. It's also important to point out that if you have a misplaced cell, it's going to maybe not behave like the rest, right? And so it's both critically important on the identity and the density of the cells to observe these properties. And this really doesn't have a direct relevance to the work we're doing, but I think it's a nice slide showing a dental plaque community, which can contain, in fact, many, many different types of bacteria. Dozens of bacteria are all thriving in your mouth right now. Many of them are fairly benign. Obviously, some of them cause cavities and decay, but they can create these very complex structures. So there are some polymicrobial, they're called corncob structures, where you have filaments of one type of bacteria that get coated with other sorts of bacteria, and they function synergistically. And how they arrange themselves are absolutely key to their behavior. All right, so here's more of dental plaque. The model systems that we're really interested in applying our technology to have been pathogenic human targeting bacteria. And one species in particular that the Whiteley Lab has been studying for a long time and is one of the world leaders on Pseudomonas group behavior is shown here, forming these biofilms or plaques inside lung alveoli. People who get cystic fibrosis, unfortunately, develop these plaques. It is often the cause of death, ultimately. And they're so damaging because they do undergo these bacterial group behaviors. So they have quorum sensing, transformation of their genes or gene gene expression, and they develop this phenomenon known as a population-dependent antibiotic resistance. So, mostly when you think about antibiotic resistance, you think about people getting some virus and getting an antibiotic prescribed, and then you genetically create resistant strains. And that's not what's happening here. This is by virtue of the bacteria existing at a high enough density to communicate with other cells to become more virulent. If you treat them with an antibiotic at that point, they're going to resist, they're not going to die. If you were to take that same colony and disperse them, they would again become susceptible. So this is all being mediated in ways that are only partially understood via various diffusible chemicals. And so what we have been trying to do is develop tools that we can use with Marvin and to understand how these different multicellular properties emerge in low cell numbers. And equally as important, the methods that we use really have to be validated. They have to ultimately survive the question of, are they accurately modeling what might happen from the dish into the lung? So, so this method that we use, I've already sort of given you the sneak preview, but basically we're using a process of multiphoton excitation, sort of schematically depicted with this Jablonski diagram. So everybody, I think, from freshman chemistry knows you can excite molecules from one electronic level to another if your photon has an energy that matches that gap. And it turns out if you have very intense light, generally that's pulsed in very short pulses, so we're talking typically femtosecond lasers, you focus that down to very small spot sizes, you can get absorption of 2, or in fact, in some cases, you can get 3 photons being absorbed simultaneously, promoting the molecule to an excited state, which then ends up, in our case, producing singlet oxygen, which mediates this protein to protein chemical interaction. Sort of molecules that we use to promote this process are going to be large organic chromophores. So something like rose bengal is a very efficient 2-photon absorber and subsequent singlet oxygen producer. So any proteins that are in proximity to the singlet oxygen can undergo this process. This is very hard to see. I think everybody probably at this point is very familiar with, say, 2-photon microscopy, but if you haven't used it or seen what it can do, this is a nice schematic that was that we took from the internet. But basically, this first microscope objective is showing excitation of fluorescein using 1-photon excitation. You can see as the laser is focused through solution, it's exciting it pretty much along the axis. Here's 2-photon excitation. Instead of using these short wavelength photons, they're using near-infrared light. And what you can't see here is this little spot. So the reason why we want to use 2-photon excitation is that the process scales as the intensity squared of the laser light. And that means that if you focus the light to a very small spot, the only place that you're going to get excitation is in a volume that's very close to the focal point of the laser. So we've got singlet oxygen being produced very close to the focal point. And then you can scan that focal plane around through solution. And you can create then 3-dimensional structures of essentially any shape that you want, anything that you could think up and specify or derive from data. You can program a scanning system to fabricate. You're going to get resolutions on the order of about a cubic micron, a little bit better laterally than axially. Typically, the lasers that we use are near-infrared lasers. So femtosecond Ti-sapphire is one of the common lasers that we use, operating at maybe 780 nanometers. So this shows the ability in real time to modify these cellular environments. These are E. coli that are scooting along a cover slip. On top of that is our solution containing bovine serum albumin and rose bengal. And what you can see my student Brian doing is basically an etch-a-sketch where he's fabricating photocrosslink BSA at the interface of the cover slip and the reagent solution. Initially, the E. coli are not sort of aware that a third dimension exists, so they're being confined by this little etch-a-sketch line. And then at the end, they're able to swim out into solution. So this just gives you a sense of the scale and of the potential biocompatibility of some of these processes.
Is there any local heating of the solution that takes place that, is going to disturb the coli or anything else here?
Yeah, that's a really good question. Typically, the powers are low enough, and the fact that the lasers are femtosecond lasers avoids most of the heating. I think Chris Hsu at Cornell has done some calculations, and under most of the conditions, it's going to be a fraction of a degree. Yeah, but it's a good point. Okay, so how do we do this? Most cases, we actually don't need really complicated structures. Most cases, we're going to make something like a box or a box inside of a box. But in some instances, you need to make things that are pretty complicated. So one way you can think about doing this, those of you who know optics may recall that if you have a light source that you focus into a plane, allow the light to defocus and then refocus it again, anything that you do to the light, patterning the light in one plane, will have a one-to-one spatial mapping into the other plane. And so one technique that my student Brian took at the time was basically to just print out a mask on a transparency. So maybe something that looked like this, multi-pronged star. You could focus then using this light into a fabrication solution. I should point out this uses a scanning system. It's going to be a raster scan, so it scans the spot in 2 dimensions. And you can refocus the light and create a shape that maps to your mask shape. And by moving then the focus to deeper and deeper positions in your reagent solution, you can make what you might generously call a 3-dimensional structure. It will have three-dimensional aspects to it. Each plane represents the thickness of your focal plane. And again, in our case, that's about a micron or a micron and a half. So you can make structures that are 10 or 20 or 30 microns tall, but they're all going to have the same shape, right? It's not going to vary along the z-axis. And if you think about wanting to make something like a box inside of a box, you're going to want to change that mask. So if you have a transparency mask, that's going to be a horrific process. I would never ask anybody to do this. There's going to be an alignment problem, right? Each new mask has to go in perfectly. So Brian, along with another student, Rex Nielsen, came up with a way to do this using an electronic mask. So this is using what's called the digital micromirror device. It's basically the heart of a computer projector. So these very small, about 15 micron diameter aluminum mirrors. And the way it works is you send an image to the projector or your DMD, and it tilts the mirrors in a certain direction so that when the laser now raster scans across the surface, your pattern is sent further down the microscope by the tilted mirrors. And those that are in, say, the part that you don't want to fabricate are in the other orientation. And so we can now create a series of aligned masks by just showing a movie on our digital micromirror device. Right? So each new fabrication design, we show a new image, and we step the fine focus of our microscope by 1 increment. And in doing that, we can make structures. This is real time. You can see the raster scan. 1 dimension is a very fast scan. The other takes however long it takes to get across this image. You can see they're making this box. There's a 10 micron scale bar. And they're showing a pattern on the DMD that looks like this. And for most of the box, we just use the same mask pattern. What you can't tell is when we get to the top, that pattern is replaced with just a square. And so that's what makes the roof at the very end. You can see about how long this takes. It's tens of seconds. One of the really great parts of using this dynamic mask approach is it's really easy to do iterative processes, to do rapid prototyping. If you make a structure and it's not the one you want, you go back and you alter your design literally in 5 minutes. And you're in business again, making a different structure. So you don't have to, go out to a foundry and that sort of stuff, or even make molds.
How long does it take to print one of these structures?
Yeah, so a single structure, again, this is a cavity that's, about 20 microns across. That was real time. So tens of seconds. So it's great for rapid prototyping. It is a serial process, right? So if you want to make something really large, or many of them, it's not a really good way to do it. And that's one of the big limitations. If you have applications where you want to make a structure that's millimeters along the side, that's going to take us a very long time to do. But you can make really fantastic structures. So these are a series of structures that my students made by making masks out of X-ray CT data. So those you can download as a stack of images, and you can make binary masks out of those, and you can make something from the Temple of Doom if you want, or alligator skulls.
So these are SEM images?
These are SEM images, yeah.
So once you have made these designs, these does not need to be stored inside of solution. You can take them out.
You can. They are hydrogels that are mainly comprised of water, and the prep for SEM is pretty sketchy. So you can get substantial collapse of structures, contraction of structures, if you don't do it, exactly right. And even there, they're going to change shape some. But we have had some luck dehydrating, lyophilizing structures too. Here are some other images. This is kind of more of the same from the last slide. You can make sections. You can see they do have internal anatomical structures. Again, they're binary masks that we've made from the data. So they're going to lose a lot of the density gradations that you might think about. But you can also design various types of structures. Here's a tip. I forget what this weapon is called. Is it a morning star? Anybody into medieval weaponry?
Yes, that's correct. Old Dungeon Master.
All right, so this is a 5-micron scale bar, and you can see the level of detail you can get, sub-micron point sizes. Here's a Mobius strip net that we've made and stapled to the surface that we designed. So really, almost arbitrary complexity is possible if you need it. Based on the type of protein you use, they're going to have various environmental responses. So in this case, we looked at 3 different proteins. BSA, our old standby, are the triangles. We actually fabricated with avidin. Those are the open squares, and with lysozyme, which are the circles. And we evaluated what we call the swelling ratio. It's not really that important, but basically it's a measure of how the size of a structure changes in our optical microscope relative to its size at pH 7. So everything's going to have a swelling ratio of 1 at pH 7. And you can see, for instance, with BSA, BSA reaches a minimum swelling ratio somewhere around 4 or 5, and it comes back up. It turns out it's very close to its isoelectric point, which is something you might guess, right? So how hydrated protein is going to depend on how charged it is. And that's a theme that works for all of these proteins. And so in this case, we made these little arcs alternating between BSA and avidin. And the movie, if it plays, is just showing you that by choosing the appropriate pH, we can selectively alter the size of these arcs. So they're attached on the ends. They're not connected in the middle. And you can see by going to these different pHs, we're able to selectively cause expansion and contraction. I'll play that one more time.
So if you have a bacteria or a fungus growing inside of this structure, it might, during growth, change the pH. Do you think that changes the structure?
Yeah, so the pH may change to a certain degree depending on the structure. We do know that it doesn't, that we can identify conditions where it doesn't increase or decrease dramatically. And we know that because cells can grow to exceedingly high densities inside the structures. Mm-hmm. So the transport through the walls is great enough. That's not generally a problem, but the pH is in the core of a large micro 3D printed structure is definitely going to get big. It's definitely going to drop a little bit at least. Okay, and this one I'm going to skip over, other than to tell you that we've used the attributes of these materials, how they change their volume when exposed to different colors of light and different types of protein, to be able to introduce cells into a chamber, cause a swelling of one type of protein material so that the chamber closes off, trapping the cells inside. We can then grow the cells. And in this particular experiment, we then have a different type of protein that we can expose with a different colored light and cause the pore to open up. And so we call this bacterial farming, basically capturing cells, growing them in one chamber, a sub-picoliter chamber to a certain density that we want, and then releasing them into a much larger chamber. We think this will be a really interesting scheme to develop bacterial group behaviors and then watch those behaviors dissipate as the cells experience a larger volume. All right, so just to give you a little bit of picture of some of what we've been doing, studying group behaviors. These environments generally are going to be pretty simple, pretty straightforward. They're not going to be Mobius strips. They're going to be boxes or boxes within boxes. And some of our early work, this was with E. coli. You can see we created the structure that Marvin actually came up with the term a lobster trap. Basically, this is a box. It's about 10 microns tall with a roof. And you can see we've got this funneled entryway into a chamber. Right, so you can see that bacteria can swim in. They can get in pretty easily, right? So they're basically funneled in. Once they get in, it's much harder for them to get out because of the funneled shape of the entryway. So in this experiment, we allowed the cells to interact with the box for 1 hour. And then there's 2 hours later. And you can see the bacteria are displaying. You can see they're moving around. They're at extremely high density. They're basically at as high of a density as you could get. They've grown, divided tremendously with generation times of tens of minutes, so appropriate for what you would expect of those cells. And they've been able to do this, again, because of the facile exchange of nutrients and waste through the walls of the container. Yes.
Was it random walk of the microbes in there?
That's a good question. We think that it was. We certainly weren't doing anything purposeful to attract them. So no chemoattractant. If you watch the extended video, it's not entirely clear that they didn't have some interest in the box, but it wasn't strategic. Yeah. I will say that, using our methods and our ability to, for instance, biotinylate this and display essentially, a wide variety of different compounds, you could strategically attract bacteria, neurons, other types of cells by, for instance, putting an enzyme on the surface that cleaves a precursor to an attractant or a repellent.
It's like 5 minutes.
Yeah, okay. All right, and this just gives you, this is kind of a jiffy pop where we let this grow for 6 more hours. You can see we got a lot of growth outside the box, but in this case, Brian defocused the microscope, and if this plays smoothly enough, you'll be able to see basically like that Jiffy Pop popcorn where the protein matrix is flexible and expandable enough that it's created a large dome that domes out above the box. All right, given the limitation on time, let me just show you one interesting result. So in this case, my student Jody fabricated these structures. Again, they're about 10 microns tall. They have a roof, and allowed Pseudomonas to enter the boxes and to grow for varying times. In one case, she allowed them to grow so that we had about 24 cells. And another experiment, she allowed them to grow for about, to develop into about 200 cells. And then she tested their susceptibility to gentamicin and used the concentration known as the minimum inhibitory concentration, basically in a flask, the concentration that will kill cells with high efficiency. And then looked to see whether they survived or not, used a live dead stain. dead is red and live is green, and was able to find sort of this tipping point for this volume of trap where a transition occurs between antibiotic susceptibility and antibiotic resistance. And you can see we had roughly 3/4 of the cells die at the low density, and very few die at the high density population. Those of you who are physical scientists might look at that and go, well, maybe the antibiotic just had difficulty diffusing through that population. We did look at that, and although there is a slight slowdown of diffusion, it would not have accounted for this difference. Okay, so just to give you one more sense for one of the developments of this technology, these lobster traps are great. They have allowed us to study simple populations of cells to grow them to densities that we want, look at form sensing development, look at antibiotic resistance development. As you can imagine, they're not so useful if your cells can't swim into the boxes. You might imagine a microfluidic environment where you flow cells and open up your entryways and allow the non-modal cells to go in, but that's challenging. What's even more difficult is if you imagine you want to, for instance, embed one type of cell within a container inside another type of cell. So complex geometries, look at core shell environments, or look at one population or one type of cell at a certain density, and then look at how a neighboring population of the same cell at a higher density impacts it. And so that's not going to be something that you're really able to do with these lobster traps. And I'll just finish up with this idea. Basically, we stole the idea from our memories of lunchtime in elementary school. Fruit cocktail and the Jello. I don't know if any of you had that in school, but basically the idea is that, you know, the pieces of fruit, they're not going to move around. As long as the fruit is still okay, you now have something that you could go into and, for instance, fabricate within and have a distribution that's going to represent a more complex situation. So think about not having fruit in Jello, but having bacteria in Jello or gelatin. And the idea there is we can suspend bacteria in our reagent. Now, not only has BSA and a photosensitizer, but it also has gelatin. You allow it to cool and gel. The cells are suspended and alive. You can then go in and fabricate a house around a cell, a single cell if we want, to grow up a clonal community, and then rinse away, heat and rinse away the reagent and grow up our community of cells. If we do that in a sequence of steps, for example, where we start with Staphylococcus, we can trap Staphylococcus in a core. We can allow it, for instance, to grow a little bit because it grows a little bit slowly. We can then add in gelatin reagent that has Pseudomonas and fabricate another box around that. And keep in mind, these are three-dimensional, they have roofs. We can then grow up these populations and examine how, for instance, having Pseudomonas around Staphylococcus influences the behavior of staph. So when we looked at this, we looked at how ampicillin affected the kill rate of staph. And so normally staph is pretty susceptible to ampicillin. It's a beta-lactam antibiotic like penicillin, right? And so with no Pseudomonas around it, we killed most of the staph. When we surrounded it by a dense shell of Pseudomonas, we retrieved a lot of survival. And our suspicion was that that was being caused by a beta-lactamase that Pseudomonas produces and displays on its membrane. So basically, the antibiotic wasn't making it through this shell. It's being degraded on its way into the staph. And we used a Pseudomonas cell line that overexpressed expressed the beta lactamase and saw a further retrieval of survivability for the staff. So it really supports that mechanism. Not an amazingly surprising mechanism, but it was, I think, a nice demonstration that we can create these communities. We can look at how complexity of the community tunes the response of different cells. And again, I apologize. There's a whole lot more I would have liked to have shown, but I know everybody has other stuff you got to get off to. So I am just going to basically say, to varying degrees, we've gone through some of the new capability set that our fabrication technology has brought to cell culture. Again, along with other tools that other people are developing as well. And I do just want to point out some of the students through the years who have contributed to this. Brian and Rex did a lot of the early work on this, as well as Jody did a lot of the initial biological studies. And then some other people who have contributed some to the slides that you saw. Marvin, who's at Georgia Tech now. But we are continuing to collaborate, and some of our other collaborators, some of you may recognize if you're from chemistry, Alan Bard. So we've done some electrochemistry studies as well. Okay, well, thanks for your time. And if you have more time, want to hang out, we can talk a little bit. So.